Now, dear students, today we shall take up the human reproductive system with special reference to the male and female. Now, we shall start with first the male reproductive system here now. In the male reproductive system, the important points which can be asked in the EAMCET, AFMC, JIPMER, AIMS, Comet K, so on and so forth, I shall put before you. Now, in the human reproductive system here now, the main components are the testes. The main components are the testes. Now, these testes are formed in the embryonic development itself, that is embryonic stage itself. In the 7th month of pregnancy, they descend into scrotal sacs. So, suppose you get a question, students, in the EAMCET or any other exam, during what time the testes from the abdominal cavity descend into scrotal sacs, should answer 7th month of pregnancy. The testes from the abdominal cavity descend into scrotal sacs through inguinal canals. Okay. Then here you must remember one important point, dear students. This can be asked under assertion reason also. In all mammals now, except a few, barring a few exceptions now, the testes are embedded in scrotal sacs. Okay, except elephants, that is proboscidea, then sea cows, that is sirenia, then cetacea, that is whales, dolphins, so except, and edenates, that is anteaters. So except these following mammals now, in all other mammals now, testes are embedded in scrotal sacs. So make a note of this point, important students now. In proboscidea is an order that includes elephants. In edenata, it is also an order that includes sloths and anteaters. Then number 3, cetacea, that includes whales and dolphins. The next, sirenia, that includes sea cows. So except these mammals now, in rest of the mammals, including human being, testes are embedded in scrotal sacs. Now in these mammals now, testes remain within the abdomen. Now this can be asked under assertion reasoning here now. Testes are embedded in scrotal sacs in human being, that could be the assertion. Then what could be the reason for that? Why are they embedded in scrotal sacs? Why do they descend into scrotal sacs through inguinal canals in 7th month of pregnancy? They descend into scrotal sacs to keep themselves away from high body temperature. Under high body temperature, even if the sperms are synthesized, they may die due to high temperature. So to avoid high temperature, in most of the mammals, barring these exceptions now, testes are embedded in scrotal sacs. In all these mammals, the 4 of them what I wrote on the board here now, in all those 4 groups of mammals now, testes are within the abdomen because of low body temperature. Because of low body temperature, testes are embedded within the abdominal cavity. In all other mammals, testes descend into scrotal sacs through inguinal canals. In human being, they descend into scrotal sacs through inguinal canals during 7th month of pregnancy. Okay. Then sometimes in human being here now, very rarely, very seldom, testes do not descend into scrotum. They remain within the abdomen even after birth. And that condition where the testes are retained within the abdominal cavity is called cryptorchidism. What is that known as? Cryptorchidism. Now this can be asked in AIMS, JIPMER and all other Comet K and all exams now. So what is cryptorchidism? Retention of testes within the abdominal cavity even after birth is called cryptorchidism. It has to be rectified by surgery, otherwise the child turns out to be sterile. Now, once the testes descend into scrotal sacs, they are attached to scrotal wall by a muscular thickening called gubernaculum. Once the testes descend into scrotal sacs, they are attached to scrotal wall by a muscular thickening called gubernaculum. So remember students, gubernaculum is a thickened structure that helps in fastening the testes to wall of the scrotum so that they don't get back once again into the abdominal cavity while jumping, hopping, so on and so forth. Now you can make out in this diagram, children, here now. Now here this is the testis. It is embedded within the scrotal sac. It descends in the scrotal sac through inguinal canal. Descends into scrotal sac through inguinal canal. Testis is attached to a scrotal wall by a muscular thickening called gubernaculum. On one side of the testis there is a muscular thickening. This muscular thickening is known as epididymis. Epididymis towards hind end gives rise to vas deferens. That vas deferens extends upwards once again into the abdominal cavity to open into the urethra. Extends upwards to open into the urethra. Now this is the inguinal canal through which the vas deferens extends upwards, and a structure called spermatic duct or spermatic cord. This can be called spermatic cord. Spermatic cord is connected to the epididymis. That is, once both the testes descend into scrotal sacs now, the testes get nourishment from the body cavity through a structure called spermatic cord. Spermatic cord consists of an artery, a vein, a lymph vessel, and a nerve fiber. So spermatic cord is the one which nourishes the testis now, and it is the only communication between the testis and the body cavity. Through spermatic cord, the nourishment is supplied to the testis constantly, because as it consists of an artery, a vein, a lymph vessel, and a nerve fiber. Now depending upon the body temperature and seasonal changes testis either they move closer to the body or they move away from the body. During winter, both the testes move closer to the body. As a result, the scrotal sac corrugates to absorb heat from the body. During summer, both the testes move away from the abdominal cavity. As a result, there is a sagging of scrotal sac during summer. So depending upon the seasonal fluctuations, there will be movement of testis within the abdominal cavity. Now dear students, now we shall talk about the internal structure of testis, the most important points with regard to the internal structure of testis. Here, if you take cross section of the testis, CS of the testis, when you observe, it is covered by an outer epithelial layer called tunica vaginalis. Epithelial layer called tunica vaginalis. This tunica vaginalis is a double layered structure. Double layered structure with outer parietal layer, inner visceral layer. With outer parietal layer, inner visceral layer. In between there is a cavity filled with the fluid, and that fluid filled cavity facilitates movement of the testis upward or downward direction. So the outermost layer of the testis is tunica vaginalis. Below the tunica vaginalis, there is a dense connective tissue sheath. There is a dense connective tissue sheath here now that forms the bulk of the testis. This dense connective tissue sheath that forms the bulk of the testis is called tunica albuginea. The dense connective tissue sheath that forms the bulk is called tunica albuginea. Now, tunica albuginea, internally it forms a single partition like this, a single partition or a septa. That median septa of tunica albuginea is called mediastinum. Now, apart from mediastinum, tunica albuginea also forms several interlobular septa. It gives rise to several interlobular septa. Apart from mediastinum, it gives rise to several interlobular septa. Like this, there are several interlobular septa which arise from the tunica albuginea and extend into the lumen of the testis. Arise from the tunica albuginea. Like that, there are several interlobular septa. Now, I shall label these students. These are interlobular septa. Now, these interlobular septa divide the testis into many lobules. As many as 250 lobules are present within the lumen of the testis. Now, in each lobule, what is this referred to as? Lobule. Now, within each lobule, about 1 to 3 seminiferous tubules are present. How many seminiferous tubules? 1 to 3 highly coiled watch spring-like structures called seminiferous tubules are present. These are all highly coiled watch spring-like structures known as seminiferous tubules. Okay, students. So, these are the seminiferous tubules here now, which are embedded within the lobule and extending towards the inner end of, like this, there are several seminiferous tubules. So, I shall label this seminiferous tubules. How many are there in each lobule? About 1 to 3 seminiferous tubules are there in each lobule here now. Now, one more important point, remember, EAMCET, JIPMER, COMET K, AFMC, AIM, so on and so forth. The most important point, within the tunica albuginea, there are certain hormone secreting cells like this. Hormone secreting cells are there within the tunica albuginea. Now, these hormone secreting cells, all these hormone secreting cells are called Leydig cells. These hormone secreting cells are called Leydig cells. So, remember, important function of Leydig cells. The Leydig cells are also called interstitial cells. Interstitial cells or Leydig cells. Now, these Leydig cells are interstitial cells under the influence of hormone ICSH. Under the influence of a hormone called ICSH, that is interstitial cell stimulating hormone. I shall repeat once again, interstitial cell stimulating hormone. It is secreted by anterior pituitary. Secreted by anterior pituitary. Now, under the influence of ICSH, the interstitial cells or Leydig cells synthesize an androgen called testosterone. They synthesize an androgen called testosterone. Now, hormonal part, we shall talk about that hormones in detail later on. So, time being, you remember, dear students, so ICSH hormone stimulates the interstitial cells or Leydig cells to secrete an androgen known as testosterone. Then, all these seminiferous tubules give rise to certain tubules, certain narrow tubule-like structures called tubuli recti. Tubuli recti. Now, these tubuli recti unite to form a network of fibers called rete testis. They give rise to network of fibers called rete testis. Now, tubuli recti towards the inner side unite to form a network like this. There is a network, dear students. So, they all unite to form a network. Here you have a meshwork. I saw there is a meshwork of fibers. This meshwork of fibers is called rete testis. I shall label this here. This meshwork of fibers is called rete testis. Rete testis. Now this rete testis in turn gives rise to vasa efferentia. In turn it gives rise to several vasa efferentia. So all these narrow tubules which emerge out from rete testis can be called vasa efferentia. And the vasa efferentia are embedded in a muscular thickening called epididymis. So all of them are embedded within a muscular thickening. This is a muscular thickening known as epididymis. So all these are vasa efferentia. All vasa efferentia, now all those narrow tubules which are embedded in epididymis are called vasa efferentia, and the muscular thickening that lies on one side is called epididymis. So this is epididymis. Now this epididymis is in turn divided into three parts. I shall label them. The most important point, once again your EAMCET point of view, the anterior part is called caput epididymis or globus major. The middle part is called corpus epididymis. The lower part is called cauda epididymis. The anterior caput epididymis, middle corpus epididymis, and the posterior cauda epididymis. Now cauda epididymis continues as vas deferens. Cauda epididymis continues as vas deferens now. Now this cauda epididymis is connected to scrotal wall by a muscular thickening known as gubernaculum. Here you find the structure called gubernaculum. So this is the gross internal structure of testis now, where the epididymis is present on one side. Now what are the functions of epididymis now? Epididymis brings about a physiological maturation of sperms. Physiological maturation of sperms. Maturation of sperms. B. Storage of sperms till ejaculation. Physiological maturation of sperms, storage of sperms till ejaculation, and they also nourish the sperms. It also nourishes sperms. So these are the functions attributed to epididymis: maturation of sperms, nourishment of sperms, and storage of sperms till ejaculation. Now we shall talk about internal structure of seminiferous tubule. Internal structure of seminiferous tubule. Now when you take the cross section of seminiferous tubule here now, within seminiferous tubule, internally it is lined by cuboidal epithelium. These are the cuboidal cells which are there all over the inner surface of seminiferous tubule. All these are the cuboidal cells which form the germinal epithelium. All these cells form germinal epithelium. All these cells form germinal epithelium. So label it, you call it germinal epithelium. Now at certain places, the germinal epithelium is interrupted by certain pillar shaped cells. These are the pillar shaped cells here now, present at certain places. These pillar like cells embedded within the primordial germ cells of germinal epithelium are called Sertoli cells. Sertoli cells. Sertoli cells are also called sustentacular cells. Now what are the functions of Sertoli cells? Number one, provide anchorage. They provide anchorage to spermatocytes and spermatids, provide anchorage to spermatocytes and spermatids. Number 2, they secrete antigen binding protein, androgen binding protein, androgen binding protein, which is also called ABP, androgen binding protein, ABP, that helps in concentration and sticking of androgen. The androgen is the hormone secreted by interstitial cells. Androgen is subjected to a kind of concentration process, or you can say a kind of dehydration process, that is brought about by ABP. At the same time, androgen is made to stick to seminiferous tubules, made to stick to the wall of the seminiferous tubules by ABP of Sertoli cells. Then apart from this, the Sertoli cells also synthesize a hormone called inhibin. So remember student, the most important point here, which can be asked in the EAMCET, JIPMAR, AFMC, COMET case, so on and so forth. They synthesize a hormone called inhibin. This inhibin suppresses excess secretion of FSH through negative feedback. Whenever there is secretion of more amount of FSH from the anti-pituitary gland, and negative feedback is sent to pituitary gland and hypothalamus through inhibin. So inhibin suppresses excess secretion of FSH through negative feedback mechanism. So these are the functions attributed to Sertoli cells. Then within this, there are several stages of sperms, Several stages of spermatogenesis are present within the lumen. They are all several stages of, like spermatogonia, spermatids, spermatocytes, like different stages of them are present within the lumen of seminiferous tubule. And remember students, each seminiferous tubule is lined by a vascular epithelium like this. There is a vascular lining which is there just below the tunica albuginea. This epithelium lies just below the tunica albuginea, encircling the seminiferous tubules. This is called tunica vasculosa. Okay, so this is with regard to internal structure of seminiferous tubule here now. Now we shall talk about the spermatogenesis process. Formation of sperms from spermatogonia is called spermatogenesis. That is, the primordial germ cells which are there within the germinal epithelium, they undergo radical changes to form mature sperms, where the entire cycle is put together, called spermatogenesis. So you have here primordial germ cells of germinal epithelium. Germinal epithelium. Now these primordial germ cells are diploid in nature. Now they proliferate, multiply to form spermatogonia. They multiply to form spermatogonia. Spermatogonia are also called sperm mother cells. They are also called sperm mother cells. They are also diploid in nature, and two kinds of spermatogonia are formed: spermatogonia A and spermatogonia B. Spermatogonia A, spermatogonia B. Spermatogonia A, spermatogonia B. Now out of these two, the spermatogonia A always act as stem cells. That is, they in turn multiply to form spermatogonia B. In turn multiply to form spermatogonia B. Spermatogonia B in turn multiply to form various stages of spermatogenesis. So they always remain within the testis throughout the lifespan of a person to synthesize sperms. So spermatogonia, whenever they multiply, they form spermatogonia A and spermatogonia B. So they never get exhausted. They remain intact and act as stem cells or raw cells. Now spermatogonia B multiply further to form various stages of spermatogenesis. Now formation of spermatogonia from primordial germ cells is called, this is the first phase of spermatogenesis, it is called multiplication phase. Multiplication phase. It is the first phase of spermatogenesis, which is called multiplication phase. Now formation of spermatogonia B from spermatogonia is called spermatocytogenesis. What is this process called? This particular phase is called spermatocytogenesis. Formation of spermatogonia B from spermatogonia or sperm mother cells is called spermatocytogenesis. Now, the spermatogonia B in turn undergo mitosis to form two primary spermatocytes. They form primary spermatocytes. They undergo mitosis to form primary spermatocytes. How do they form primary spermatocytes now? The spermatogonia grow at the expense of the remaining spermatogonia and thereby increase in their size to form primary spermatocytes. Since this process involves growth and enlargement of spermatogonia to form spermatocytes, this is also called growth phase. What is this also known as? Growth phase. So second phase of spermatogenesis is growth phase, which involves formation of primary spermatocytes from spermatogonia. Now these primary spermatocytes are also diploid because it is only a kind of mitotic division, so they are also diploid in nature. Now these primary spermatocytes undergo meiosis. They undergo meiosis. Now primary spermatocytes undergo first phase of meiosis, that is reduction division, meiosis 1, to form two haploid secondary spermatocytes. So they undergo meiosis, first phase of meiosis, to form secondary spermatocytes which are haploid here. Now the secondary spermatocytes undergo second phase of meiosis to form spermatids. Here I shall depict the entire process and show you students. Here you have primordial germ cells. They multiply to form spermatogonia. Spermatogonia are also called sperm mother cells. Spermatogonia are diploid in nature. Formation of spermatogonia from primordial germ cells is called multiplication phase. Now the spermatogonia, as I said earlier, A and B are there. Formation of A and B from the spermatogonia, that is from primordial germ cells, can be called spermatocytogenesis. Now B always multiply to form various stages. A remain intact. B cells always multiply to form various stages of spermatogenesis here now. Now the spermatogonia increase in their size, grow at the expense of the remaining to form primary spermatocytes. Which are also diploid. Formation of primary spermatocyte from spermatogonia is called growth phase. Is called growth phase. Now primary spermatocytes undergo meiosis, which is reduction division, which is also called meiosis 1, to form two haploid secondary spermatocytes, Now each secondary spermatocyte undergoes second phase of meiosis, that is equal to mitosis, to form spermatids. Four spermatids are formed from each primary spermatocyte. Single primary spermatocyte divides to form two secondary spermatocytes. Each secondary spermatocyte divides to form two spermatids. So altogether 4 spermatids are formed from single primary spermatocyte. Now the meiosis 1 and meiosis 2 are put together called maturation phase. Maturation phase. Now each spermatid undergoes certain physiological and morphological changes to form 4 mature sperms. Then 4 sperms are formed for each spermatogenetic cycle. So remember students, now the most important point, EAMCET point to another exam point of view, how many sperms are synthesized for each spermatogenetic cycle? You must answer 4. 4 from each primary spermatocyte, and they are all haploid. Transformation of spermatids into sperms is called spermiogenesis. Or spermateliosis. Is called spermiogenesis or spermateliosis. Formation of sperms from spermatids is called spermiogenesis or spermateliosis. So all these, how many phases are there in the entire spermatogenetic cycle now? Number 1, multiplication phase. Number 2, growth phase. Number 3, maturation phase. Number 4, spermiogenesis or spermateliosis. Now these sperms remain embedded within the seminiferous tubules. The process of detachment of sperms from seminiferous tubules to set them free is called spermiation. So this is another important point. Remember students, EAMCET and all other exam point of view, spermiation is the process of detachment of mature sperms from seminiferous tubules to set them free so that they are sent out along with the seminal fluid in the form of semen is called spermiation. The process of detachment of sperms and their liberation in the form of semen is called spermiation. Now dear student, now we shall talk about the structure of sperm. So far you have learnt about the internal structure of testis, then organization of male reproductive system, then internal structure of seminiferous tubule, then the entire spermatogenesis process, the various phases of spermatogenesis, and now we shall focus on the structure of a sperm. Now the sperm basically contains head, neck, middle piece, and tail. So these are the four major parts of the sperm, head, neck, middle piece and tail. Now the head comprises of a cylindrical nucleus. It comprises of a cylindrical nucleus. A rod-shaped cylindrical nucleus is present within the head. Now the head consists of the anteriorly placed protuberance, small protuberance known as acrosome. Anteriorly placed small protuberance known as acrosome. Remember students, remember the most important point here now, which was asked earlier in several exams. Now you may get even this time also, that is acrosome. Which cell organelle gives rise to acrosome? Should answer Golgi complex. So acrosome is a derivative of Golgi complex. Now what are the functions of acrosome? The acrosome synthesizes certain enzymes like A. Hyaluronic acid, number 1, hyaluronic acid, hyaluronidase enzyme that damages the hyaluronic acid. Hyaluronic acid is there within the cortical cells of the ova, so it helps in breaking open those barrier while penetrating through the cortical cells. Hyaluronidase enzyme, number 1. Number 2, corona penetrating enzyme. Corona penetrating enzyme helps in piercing through the corona radiata of ovum. Number 3, acrosin. Acrosin is also called zona lysin. That is, it helps in penetrating through the zona pellucida of ovum. The ovum is surrounded by inner non-cellular gelatinous sheath called zona pellucida and outer a radially striated layer called corona radiata. So this enzyme helps in penetrating of the sperm through corona radiata, and this particular enzyme helps in penetration of the sperm through zona pellucida. Okay, apart from this, the outer plasma membrane of the sperm head is deposited with certain proteins, cholesterol, so on and so forth. The process of deposition of various proteins, phospholipids and cholesterol within the outer plasma membrane is called decapacitation. So remember student, the most important concept here now, the outer plasma membrane of the sperm head is deposited with phospholipids, proteins and cholesterols. The phenomena of various substances being deposited in the outer plasma membrane of the sperm head is called decapacitation. So at the time of fertilization, all of them are lost. Proteins, phospholipids and cholesterol are lost, and thereby they make the sperm very active so that it could easily penetrate into the ovum. The process of loss of all these substances to make the sperm very agile and enabling it to fuse with the ovum is called capacitation process. So it is subjected to capacitation process at the time of fertilization. Sperm is subjected to capacitation process at the time of fertilization. Capacitation involves loss of cholesterol, phospholipids and proteins from the outer cell membrane of the sperm head. Okay. So with regard to acrosome. Then if we take the neck here now, the neck contains a centriole. This is the proximal centriole. The proximal centriole helps in the cell division of the zygote. It gives rise to spindle fibers during the cell division of the zygote. Then middle piece contains clusters of mitochondria. Several mitochondria are embedded in the form of a cluster in the middle piece. The cluster of mitochondria is called is called Nebenkerin. Okay, the cluster of mitochondria embedded within the middle piece is called Nebenkerin. Remember the most important point. Then towards the hind end of the middle piece there is another centriole which is called distal centriole. The distal centriole gives rise to axoneme. It gives rise to axoneme or axial fiber. It gives rise to axoneme or axial fiber that provides strength and fragility to the tail here now. It provides strength and you can say the vibratal movement of the tail here now. Now the tail has got two pieces, main piece and end piece. Main piece of the tail is internally strengthened by the axoneme. The end piece also contains axoneme but does not contain outer cytoplasmic sheath. That is, the axoneme extends beyond the main piece in the form of a naked structure which can be called end piece. So the tail has the main piece where the axoneme is ensheathed by the outer cytoplasmic layer, and the axoneme extends beyond the main piece in the form of a naked filament known as end piece. So this is the broad structure of the sperm. So remember important points with regard to the function of axoneme, then function of proximal centriole, function of distal centriole, and Nebenkerin, that is mitochondrial cloud or mitochondrial spiral that lies within the middle piece. Mitochondrial spiral that lies within the middle piece. Now you have learnt about the epididymis, the mode of formation of vas deferens now. Now look at these students. This is the testis. Testis on one side, it has got epididymis. The epididymis gives rise to vas deferens. This is the vas deferens that extends upwards. That extends upwards into the abdominal cavity through inguinal canal. Then it loops over the urinary bladder. That vas deferens loops over the urinary bladder and unites with the duct that emerges seminal vesicle. This is vas deferens. Here you have seminal vesicle to form an ejaculatory duct. Okay. Once it unites with the duct of seminal vesicle, it's no longer called vas deferens, it is known as ejaculatory duct. Now that ejaculatory duct unites with ejaculatory duct unites with the neck of urinary bladder. This is the neck of urinary bladder. So ejaculatory duct unites with the neck of urinary bladder to form urethra. And now this urethra opens out through penis. Okay, it opens to exterior through penis. So like this you have here now. Look at the students here now. So this is the vas deferens that extends upwards, unites with the duct of seminal vesicle to form ejaculatory duct. Ixacle duct unites with the neck of urinary bladder to form urethra, and urethra opens out through penis. Now this urethra, it has got three parts now. A part of the urethra is embedded within the prostate gland. Here you have your prostate gland here now. This is the prostate gland. This is called prostatic urethra. And a part of the urethra is naked. This is the naked part of the urethra, which is called membranous urethra. And a part of urethra is embedded in the penis. This is called penile urethra. Penile urethra. Now the urethra is divided into three parts now. Prostatic urethra that traverses through the prostate gland. Then you have membranous urethra, which is naked. Then penile urethra, which is embedded within the penis here now. Now, after emerging through the prostate gland, it communicates with another reproductive gland, which is called bulbourethral gland. Communicates with another reproductive gland called bulbourethral gland. So this is with regard to the organization of the ducts within the male reproductive system here now. So remember students, the three important glands are the seminal vesicle, prostate gland and bulbourethral gland here now. Now we shall talk about the functions of all those three glands. Now the seminal vesicle secretes an alkaline fluid called seminal fluid, which is seminal fluid is alkaline nature, and the seminal fluid consists of a prostate gland, that is prostaglandins. It consists of number 1 prostaglandins, number 2 inositol, number 3 fructose, number 4 citric acid, number 5 coagulating factors. So it consists of prostaglandins, inositol, fructose, citric acid and coagulating factors. Now what is the function of prostaglandin? Prostaglandins induce contractions in the female genital tract, to facilitate ascent of sperms after ejaculation. After ejaculation or insemination, sperms are made to ascend through the genital tract to reach the fallopian tube by the contractions of the uterine wall, contractions of the vaginal wall, and those contractions are triggered by the prostaglandins of seminal fluid. So prostaglandins induce contractions in the female genital tract to facilitate ascent of sperms after insemination. The students, inositol, fructose and citric acid, all three of them provide energy. All three of them provide energy for motility of the sperms. They provide energy for the motility of sperms, movement of the sperms. Coagulating factor helps in coagulation of the semen soon after ejaculation. It helps in coagulation of semen, that is solidification of semen soon after ejaculation. So with regard to composition of seminal fluid. And remember students, this alkaline nature, another important point of seminal fluid, the alkaline nature of the seminal fluid helps in neutralizing the acidity of urethra of female after insemination. Helps in neutralizing the acidity of urethra of female after insemination. At the same time, it helps in neutralizing the acidity of urethra even of male also. Even the male, when the urine is made to pass to the urethra, the entire urethra becomes acidic. To the same urethra, if the semen is sent out, because of acidic nature, the sperms may die. So at the time of ejaculation here now, the entire urethra is made neutral by the alkaline nature of seminal fluid and alkaline nature of prostatic juice. And remember students, one more important point here at this juncture, fructose has forensic importance in the medical field here now, because the fructose is usually secreted within the alimentary canal of human being here now. Okay, fructose is a kind of say disaccharide here now. It is digested at the time of digestion here now. Okay, whereas outside the human body, the only source of fructose is the semen here now. That is, when there is a kind of sexual assault here now, suppose traces of fructose are found on the body of the woman, it could be confirmed that the woman has subjected to sexual assault. So it has got the forensic importance in the medical field here now. So outside the human body, if the traces of fructose are found, it could be because of the sexual assault here now. Okay, so that is significance of the fructose in the human body, apart from providing energy to the sperms and taking part in the process of digestion. Now prostate gland. Prostate gland is there just below the seminal vesicle here now. Okay, it secretes prostatic juice. Prostatic juice is also alkaline in nature. It is also alkaline in nature, and prostatic juice consists of calcium phosphate Ca3PO4 taken twice, and it consists of acid phosphatase enzyme, calcium phosphate acid phosphatase enzyme. Then it also contains prostaglandins. It also contains prostaglandins. Okay. Then it also contains amylase enzyme. Then it also consists of prefibrinolysin and prostatic specific antigen PSA. Acid phosphatase enzyme, calcium phosphate, prostaglandins, amylase and prefibrinolysin and prostatic specific antigen PSA. This prefibrinolysin and prostatic specific antigen, both of them help in dissolving the semen after insemination. That is, secretion of seminal fluid helps in solidification so that all the sperms are held together. They are prevented from being scattered here and there here now. Then to ensure fertilization process, the sperm should be set free from the semen here now. The sperms are set free because of this effect of prefibrinolysin and prostatic specific antigen PSA. So with regard to composition of prostate gland. Next you have Cowper's gland. Cowper's gland is also called bulbourethral gland. The Cowper's gland secretion helps in neutralizing the acidity of urethra. Neutralizing acidity of urethra of male and even that of female after insemination. It helps in neutralizing the acidity of male and that of female after insemination. That is a function of Cowper's gland or bulbourethral gland. We shall talk about the structure of penis here now. Penis is covered by an outer loosely arranged skinny sheath here now. The outer loosely arranged sheath of the penis is called foreskin or prepuce, and the inner part of the penis is called glans penis. Inner part of the penis is called glans penis. Now what is the structure of glans penis here now? Look at this. If we take the structure of glans penis, It contains 2 columns of tissues on dorsal side, a single column of tissue on the ventral side. The 2 columns of tissues which are present dorsally are called corpora cavernosa, and a column of tissue that lies on the ventral side is called corpus spongiosum. It's called corpus spongiosum. The corpus spongiosum encloses urethra. It encloses urethra like this, and there is a tissue that forms the corpus spongiosum. Then within the corpora cavernosa, numerous blood vessels are present. Arteries and veins are embedded together. Several arteries and veins are embedded together, apart from nerve fibers embedded within the corpora cavernosa. And corpora cavernosa is said to be an erectile tissue within the penis, responsible for erection of penis at the time of intercourse. Okay, responsible for orgasm or erection of penis at the time of intercourse. So remember, students, get a question, which tissue is considered as the erectile tissue of penis? You should answer corpora cavernosa. So there are 2 columns of corpora cavernosa and a single column of corpus spongiosum. Corpus spongiosum encloses urethra, and these are the muscle fibers which help in holding all these 3 columns of tissues together. And the entire penis is covered by integument. As I said earlier, as I made a mention earlier, this is the integument that covers the entire penis. Integument or foreskin. And there are certain glands within the foreskin. The glands present within the foreskin, they secrete a white powdery substance called smegma, and those glands are called Tyson glands. Tyson glands are present below the foreskin, and they secrete white powdery substance called sigma.